Most of hand sanitizer and disinfectant in the market, they contain alcohol or benzalkonium chloride as the active ingredient to kill the germs. Our product, which is non-alcoholic hand sanitizer and disinfectant, are made from 100% food-grade ingredients, thus give a minimum toxicity to the skin. Our formulation is water-based, made from 3 main ingredients, which plum extract, peroleol, and non-ionic surfactant. Our lab testing also showed that the formulation exhibited good antioxidant, which comparable to vitamin C and vitamin E.
We know that this product is meant to treat a superbug. Superbug means bacteria that have resistance towards common antibiotics. And we know that every day we have seen a lot of cases, patients with superbugs, they cannot be treated and they just die because of the complication. With that in our mind, so we believe our product has a significant role in combating those superbugs. So we also tested this solution on several surfaces, mimicking hospital environment. For instance, we used a stethoscope, where the doctor wears the stethoscope and check the patient, examine the patient, and also use strainer, that also mimics the things that can be found in the hospital. And we also use so-called the ladle, the surfaces mimicking like the back rail in the hospital. So because they are made of steel, if not mistaken, So and the last we use cutting board, so to represent where people use cutting board for food processing and whatnot. So we use that surfaces, and surprisingly, at 5 seconds, this product, our product, can actually kill all the bacteria. At 5 seconds. And how do we know that? We have the experiment where we put the bacteria together with the control, everything, and with the solution. And we put that solution and we try to isolate the bacteria again, try to sample that surfaces. And we try to bring back to the lab and try to grow them on the media, and surprisingly no bacteria can be found on the media. So meaning that the solution at that particular time, at 5 seconds, very fast, will be able to kill 99.9% of bacteria.
We would like to test the anti-coronavirus properties of our product. However, due to strict requirement to grow the SARS-CoV-2, we decided to use another beta coronavirus, which is in the same group with SARS-CoV-2. Human coronavirus OC43 is our choice for this preliminary testing. The result from our preliminary testing show our product has some degree of antiviral activity.
Upper left panel show highly cytotoxic effects on cells where cells infected with virus became recollected, and this is an indication of virus infection. Lower left panel show when the product was added, it prevents some viruses infecting the cells, therefore the number of recollected cells was reduced. This shows the product has some degree of antiviral activity. In silico molecular docking study of the active ingredient in the formula, which is the monoterpenes, with 3 target proteins include SARS-CoV-2 main protease and replication protein. Second, SARS-CoV-2 RNA-dependent RNA polymerase. Thirdly, the binding domain of the SARS-CoV-2 spike protein. The in silico computer simulation shows that the active ingredient can interact with all those proteins.
Some disinfectants are toxic, some stain, corrosive, and have unpleasant odor. Our product will not damage any surface it is intended for. Simple corrosive studies show that no corrosion effects on metal. Tests also show that the product does not leave any stain on fabric and mask. The production of our nano-emulsion formulation is using a green synthesis method. The technique conducted is by mixing all the ingredients simultaneously. The method is simple, affordable, and easy to handle. Our product has lemon scent, which eliminates the odor and leaves the room fresh and bright.